Welcome to the last part 5 of January 2023. Please make sure to subscribe to make sure that you get notifications of when I upload videos moving forward. Let's get started. Hello students, welcome to the last video that I'm posting before the June 2023 exam. Some of you waited till the last minute, to watch my videos. It's okay, you're here, and any time before the exam is a good time, okay? So you made it. I want to talk about a couple of things before we start the part D, the last part of the exam, and I want to show you the videos. So part 1, I know I have more than this, but you see how I have 5.9 thousand, and then part 2 is 1.7. And then part 3, part 3 is, it got cut off, but it's like 700. And then part 4, we have here 522. This means that the people who watched the first part did not go to part 2. A lot of people did not go to part 2, and then less people to part 3, and then less people for part 4. And then I have no idea from now until the test tomorrow how many people are gonna watch my last video, but that's not good. Like, watch all of it, watch all the videos so that you can have an entire exam under your belt. Okay, that's super important, especially this part D. Okay, tomorrow, Wednesday, the 14th, is your exam. Not this one, not English language arts, living environment. Your test is in the afternoon, okay? So you have the morning to relax, study a little bit if you need to, but I want to talk about how you prepare for tomorrow's exam, okay? If this is not June 2023, or if you're not about to have the test, please go ahead and skip this and go straight to me going over the questions if you'd like, or not, or listen to this, because this is true for every single Regents exam that you take. Okay, so this is just advice on the best practices for taking this exam. So let's start with what to bring. Okay, what to bring for the exam. Let me change my color here. So I know that a lot of schools are going to give you the materials, but you can't trust their stuff. Raise your hand, a visual, a mental hand. If you have if you got supplies from the school and it was just the crappiest pen you've ever gotten, the crappiest erasers, the crappiest pencils, schools try to do their best, okay? Schools are always trying to be there for you. So I'm not knocking the schools, but you have to bring the best of the best for yourself. And the best of the best can be located in the 99-cent store. Have you gotten the mechanical pencils from the 99-cent stores? I'm not talking about the mechanical pencils that are fancy and you click. I'm talking about, they're like this, and then they keep going and going and going, and then it's like encased. So then when this graphite runs out, you can take this off and then put it back here, and then the next one is going to have a sharp one. That is the best. I love those. You can get a pack of 12 for, I don't think it's $1 anymore. I think it's like $1.50. The dollar store is not 99 cents anymore. You know, inflation. So to bring 2 mechanical pencils with a trustworthy eraser. Make sure that when that eraser erases, it is good. So test it out. Okay, make sure you have that. Oh no. Okay, it's back. Okay. Whew. Okay, next. 2 black or blue ink pens. Not pink, not purple, not green. Regular plain black or regular plain blue. You see this blue on this text? That's too colorful. I'm talking about the boring, regular academic pens. Do not get a cheap pen. Get a gel pen. Why? Because it's smooth. And those really cheap big pens is the reason why when you write, your wrist starts hurting. When you're taking a long test and you write and write and write and write, and your wrist starts hurting, it's because of the quality of the pen. Okay, so gel pens are the smoothest. So, try to get yourself a gel pen if you don't have that already. Number 3, non-graphing calculator. You are supposed to get a non-graphing calculator. These are the calculators that looks like this. Not your graphing calculator. You're not allowed to have that because that stores notes. You can store notes in there in a whole textbook and they're not going to allow it. Okay? So non-graphing basic Hello Kitty basic calculator that has plus, minus, division, and multiplication. That's it. That's all. That's the non-graphing calculators. A ruler with no additional writings on it. Some rulers have reference notes on them, like a centimeter, how much centimeters equals a foot, an inch, how many inches equal a foot, things like that. You get a plain regular ruler. That's for the graph. Remember that you need that to make those perfect lines. A bottle of water with ice. Hook it up. Hook it up. Listen, I'm an ice chewer. I love chewing ice. It's like a thing. And I love cold water. If you're someone that actually doesn't like cold water, then this doesn't affect you. But if you are someone that gets thirsty, or that you're going to be in a hot room, or this can keep you awake, please do get a bottle of water and put ice in there. And hopefully you have a really good bottle of water that it can stay really, really cold and the ice doesn't melt for hours. Make sure you have snacks on you, like a granola bar or peanuts, a dry snack, not like a bacon, egg, and cheese for my New York City kids, or wherever you are in New York State. Dry snacks so it packs well. You're not worried of it spilling. If you're about to take the test, let's say that your school has you guys taking the test at 12:30 or 1 p.m. A half hour before, if you're starving, eat your snack. I don't think you can eat during the exam. It just depends on your school. Maybe, you can ask the teacher, can I step outside real quick? And then you eat your snack. I mean, I know I would allow it. I wouldn't have a problem if I'm proctoring an exam, but every school and every teacher's different. So you don't want to bring any attention to yourself. Just have the snack right before. And then you should be good. You shouldn't get hungry again. And then you'll also have the bottle of water. And then keep the bottle, if you do have a bottle of water, keep it on the floor. Don't keep it up with you on the desk, because if that it could fall on your test and your test can get wet. So you don't want that. Make sure to drink water in the morning and stay hydrated, but don't overdo it. So when you wake up in the morning, have a nice glass of water. Okay? I'm not saying chug a bunch of water, because then you're going to use the bathroom a lot. So, yeah, make sure that you are hydrated. Okay, things that you should not do. Do not do this tomorrow. Do not drink energy drinks. It's full of sugar and caffeine, and you're going to crash. It's not good for a 3-hour exam. You're going to feel fine for the moment, but then in the middle of the exam to the end, you're going to get sleepy, tired. You're going to have a hard time keeping your eyes open. Do not do the energy drinks, okay? Do not drink coffee if you know it makes you poop. Do not drink coffee if you know that it gives you a stomach ache. Don't do it. Do not eat dairy, meaning anything with milk, if it makes you poop or if it gives you a stomach ache. Do not eat candy. Do not go into the store and buy candy. It will also make you crash like the energy drink. Do not eat potatoes. Don't have French fries for lunch. Don't eat potatoes. This, or any kind of potato. This makes you sleepy. Potatoes have a natural chemical in them that makes you sleepy. Do not go to bed at 4 a.m. studying. This time around, your test is in the afternoon, so you can study a little bit more in the morning if you want. But go to bed at a decent time and get 7 to 8 hours. If you do not do this, you might be the type of student that's going to fall asleep during the test, and you're not going to concentrate. And all the hard work that you did throughout the year and watching videos and practicing, it's going to go to waste because you're going to be so tired that you can't concentrate. Wake up and watch more videos. Review a little. Do not overwatch videos of any kind the day of the exam. Your eyes are going to get super, super tired and red, and then your eyes are not going to work for the exam, okay? So don't overdo it with screen time in the morning of the exam. Okay, so those are my tips and tricks. Now let's talk about Part D. Part D is all about the 4 labs. I'm going to go through the 4 labs super quickly so that you are reminded of what you did during the year. Do you see the screen right now? Do you recognize this? If you do not recognize this, you did not do this laboratory experiment. If you didn't do this laboratory experiment, that is not a good thing. And if you don't recognize it, the only thing I can say is that you were absent for like 2 to 3 days around this time and you never made it up. And the school didn't check that you did this. You cannot take the Regents unless you finished all 4 of the following. This is diffusion through a membrane lab, okay? You were focusing on diffusion. Diffusion is the movement of molecules across the cell membrane from an area of high concentration to an area of low concentration, okay? Active transport is the opposite. But this was experimenting on diffusion. This was when you took a plastic looking bag, right? It looked like a plastic bag, and you put here glucose, which is sugar, and it should have been in the form of a liquid and it was clear. And then you put starch. And then you tied each end of this. So it looked like a milky substance in this bag. And then here in the water, and this is the beaker. This is a beaker, okay? In the water, you put something called iodine, which is a starch indicator. That means that as soon as it touches starch, it turns dark, like a dark blue or a black color. So then time passed, you did this. Time passed about 20 minutes. And then, do you remember the bag looking, let me pick a color that mostly resembles what you saw. So then you saw it turning like this, right? You saw it turning a dark inky color. What happened was that iodine then diffused into the bag, and then the starch stayed here in the bag. The iodine reacted with the starch and then started turning this color. Overnight, the whole bag should have been dark because then the iodine mixed with the starch. And as soon as iodine touches starch, it turns a dark color, meaning, oh, this is positive for starch. And then the glucose was then found in the water. Well, how did we know? You should have taken this water, sample of this water, right? And you put it inside a test tube, right? And then you heated it up. And then this turned, if it had glucose, it turned like some kind of yellow-orange color. So it was positive for glucose. So here is the end result of this lab. You started with starch, glucose in the bag, and iodine in the water. After 20 minutes, you then had iodine in the bag, starch stayed in the bag, and then glucose was in the water. Why did starch not diffuse out? because of the size. It was too big to fit through the holes of the bag. Why did glucose diffuse out of the bag? Because of the size. It was small enough to go through. That was it. That's it. That's the exam. I mean, the lab. Nothing else. Here, there was a question on, you have S for starch, you have I for iodine, and then G for glucose. They gave you the I's in the initial one. Okay, yeah, we put iodine in the water outside, and then we put glucose and starch on the inside. Then they're like, okay, what happened at the final stage? No problem. The iodine is now in, the starch stayed, and then the glucose was out in the water, period. That's it. Then it was the osmosis lab. This is diffusion of water. This is water going from high concentration to low concentration. Okay, that's it. It's the diffusion of water, osmosis. And then what you were supposed to do, you were supposed to take a red onion and look at it under the microscope and then draw and see what you found. Okay, so a red onion was really purple, but they call it the red onion. So based on your observations, draw and color a typical red onion cell mounted in water. Okay, so you should have drawn the cell wall, the cell membrane, and a little dot that was the nucleus. Okay, then you added salt. You took a solution, okay, and you added it on this side of the cover sheet, the cover slide. You put paper towel on the other side at the other end, and then that solution went this way so that you were able to put the salt on the onion cell, right? So then they're like, okay, what does it look like now after the salt solution? The cell wall stays intact. Let me make this bigger. But the cell membrane shrank. It shrank. Why? Why did that happen? Because salt diffused in and water diffused out. But the cell wall stayed the same. It didn't shrink. The cell membrane did shrink. Okay? Because it had to make room for the salt. So then what does that look like? What should your drawing have looked like? So your drawing here should have been the cell wall and the shrunken cell membrane. And then to label, cell wall, cell membrane is this outermost layer. Okay. And that was it. All of these extra questions, everything else, no, not necessary, because this was the main thing that you needed to learn from the diffusion through a membrane lab. Second lab that you should have done. I'm praying that you're like, yep, did the first one. Yep, Miss T, I already know. I already know what you're going to say about making connections. Making connections was a lab of you getting data and finding a connection. Like I got all these numbers from an experiment that I'm doing. Now I know what's happening. I know what I can conclude. So what you did was you took your pulse 3 times and you got numbers for each time. And you did it for 20-second intervals. So you did trial 1, 20 seconds. Let's say you got 21. Let me make this bigger. You counted 21 pulses. Trial 2, again, you got about 20. Trial 3 again, and you got about 19. This is you taking your pulse right here, and every boom, 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 boom from your heart that you felt, you counted it in your head for 20 seconds. And that's going to give you a number. But you're trying to see how much your heart beats per minute, and per minute is 60 seconds, which is why you have to multiply by 3, because 20 plus 20 plus 20, 3 20s gives you a 60. So to save time, you take your pulse for 21 seconds and then you multiply by 3, and that gives you the per minute. Okay? This is the part where if you're asked this on the exam and you get nervous and you can't do multiplication, you have your trusty calculator. You are supposed to have a calculator, as I said before in the beginning of this video, unless you skipped it. So 21 times 3 is 63. 20 times 3 is 60. 19 times 3 is 57. Okay. But this is the trial. And you are to get the average pulse rate. Well, how do you take an average? You take all of the numbers and add them. And then you divide them by the number of trials. Okay? So let's add them. 63 plus 60 plus 57 equals, so let's see what that gives us first. Okay? 63 plus 60 plus 57 gives us 180. Now pause. Now you take that number and you divide it by 3. Divide it by 3. Why 3, Miss T? 3 because you have 3 trials. Okay? So 180 divided by 3 is 60. So your average pulse rate was 60. This was just a sample. You know, I just randomly got these numbers of what's also realistic. The normal beats per minute for a human is from 50 to 100. Okay. So you got your average pulse rate, and then your teacher was supposed to collect everyone's pulse rate, and then you were supposed to make a bar graph dependent on that. And we're not going to do that because this is just reminding you what the labs were for. Then the second part of this lab was to test for something called muscle fatigue. Muscle fatigue is when your muscles hurt after exercising for a very, very long time. The reason why your muscles hurt for after exercising for a long time is because your muscles create what's called lactic acid. That lactic acid is what creates the soreness in your muscle after you've exercised a certain part of your body for a very, very long time. So here it wanted to test your muscle fatigue. Okay. You took a clothespin and you open and close it as fast as you can for a minute. And then let's say the first try you got 100 times, you were able to open it. Then you did it for a second time. And let's say the second time you got 90, you got less. Well, then you had questions like, why do you think that is? Oh, I got less the second time because I probably developed lactic acid and then it got sore. Well, let's say it was the other way around. What if you got 90 the first time and 100 the second time? You could have said that you didn't produce lactic acid. Or that you had a genetic, you genetically just are stronger, or whatever the case may be. But that was it. That was the second part. And then you had some questions to answer and you did it. Good. If you didn't do it, you were probably absent, or your teacher, you didn't do it. Okay. Third one was Beaks of Finches. This one's a really fun one. I go crazy with this one with my students. This one is so much fun. You become this cute guy, this cute little finch. You become this bird and you are competing for food. And you did 3 trials. Okay. You were given beans and it was you and then a partner. And then you competed for food and you were supposed to have like at least 13 seeds that you ate, and then you were screaming at your partner, why aren't you fast enough, blah, blah, blah. And then you would get an average, right? So you collected all of this and you were supposed to have a 13 or more to survive and go to the next island. So this was testing natural selection, survival of the fittest. And you were supposed to choose a specific beak. It could have been a spoon, a fork, chopsticks, whatever your teacher had out for you. And that was dependent on then, how well you did. The best one was like the spoon or using your hand a specific way. And the worst ones was the knife and the fork for obvious reasons. And the chopsticks were horrible if you don't know how to use chopsticks. So, and then you just answered questions pertaining to your results and the class results. And then you had this wheel here, and this wheel is so easy to interpret. And hopefully we can practice some questions with that. And that was it. That was the lab. The last lab you should have done for the year is called Relationships and Biodiversity. This was lab number 4. This was a big one. You were supposed to do, let me see if this one has it. Yeah. Let me see if I could flip this. Yeah, rotate this page. Okay. So you were supposed to check for, you had a Botanicurus, right? This is a plant that had the cure for cancer, and it's going extinct. So you were supposed to see if species X, Y, and Z are closely related to this, so that if this goes extinct, then the other one can take over. So first you looked at the leaves, you compared the seeds, and then you compared the stems. Then the molecular evidence, you compared chromatography, you compared an enzyme, if it fizzed or no fizz. You compared the DNA, the strips, the protein synthesis strips, okay, which is actually this one. And then this one was the protein synthesis amino acid sequences, which was this guy over here. And then you had to glue and paste strips where they belonged. And the winner was species Z. Species Z was the one that was closely related to, closely related to Botanicurus. Botanicurus was closely related to species Z. So then for this one, number 6, the best answer would have been this one. This is like just an ancestral graph that shows you that species... Sorry, let me go back to that. Oh, that was weird. Species Z and BC have a common ancestor because they had most similarities. Species X and Y had a common ancestor too, because they had a lot of differences, but similarities, but then they all had a common ancestry because they had a lot of similarities between each other. But this one's the most accurate because Botanicurus and Z were had the most in common. And I'm not going to go through it because that's a whole video. That's a whole, like, probably 2-hour video, 2-part video right there. Okay, guys, let's start the review for part D. If any of you skipped the first, 10-15 minutes of this video, I really encourage you to watch it. But I understand you just want to get to the question, so let's get started. This is the last part. This is it. I want to wish you guys so much luck tomorrow for June 14, 2023 exam. You got this. You made it to my video. I think this video is only going to have like 100 views around this time because people are not following through. But anyways, you're here, so you're a superstar. Okay, so let's get started. Let's finish his exam. Directions 73 to 85. For those questions that are multiple choice, record on the separate answer sheet that looks like the bubble sheet when you get it the number of the choice that of the given best completes each statement or answers each question. For all other questions in this part, follow the directions given and record your answers on the spaces provided in the examination booklet. I cannot stress to you enough, I don't know why this test does this. But you see how this is a multiple choice. This is a multiple choice. Multiple choice. You have to make sure that you transfer the correct answers to that bubble sheet in the correct number. Because then it goes back to short responses that you put right on this booklet. Okay? So be very, very careful with your transferring of answers. Okay, so let's get started. This one is already looking like it's the relationships and biodiversity lab. Note that answers to question 73 should be recorded on the separate answer sheet. Okay, let's take a look and read. Base your answer to question 73 on the information and illustrations below and on your knowledge of biology. The illustrations represent cross sections of 2 different plant stems. So I'm going to annotate I'm going to annotate plant stems. Okay. A student compared two stem cross sections. The stem cross section A is from a plant that can be used to produce products with valuable medicinal properties like botanicures, from what I said before. Stem cross section B is from a plant growing in the same area of the forest, and its usefulness for providing medicines is unknown. The student concluded that the stem cross section had many structural similarities and that the plant that produced cross section B would produce the same valuable medicinal products. Okay, so I'm going to annotate the student's conclusion, saying that plant B is able to produce the medicinal properties based off of just this physical characteristic. Is the student's conclusion valid? Absolutely not. I don't even have to read this, the choices, because no, you don't know if B makes the medicine just by the way it looks. You have to check for the DNA. You have to check for the chemical enzyme if it's there. You got to check for other things, for the proteins that it produces from the protein synthesis. So we have to choose an answer that says that there is not enough information. Well, we're definitely going to cross out, the yes, because the conclusion is not valid. So let's take a look at the first no. No, because he did not evaluate soil conditions. No one cares about the soil. No, because this structural evidence alone is insufficient and molecular evidence should be obtained. Yes, 74 is 4. Okay, let's take a look at the next one. Number 74. A student was jumping rope and noticed that her heart was beating faster. The student's heart rate probably increased as a result of when you are exercising, your muscle cells need more oxygen. So your heart beats faster. That's it. When you move faster, you need more oxygen to your muscles and your heart beats faster. Super, super, super simple. Okay, so let's see what our choices are. A decrease in the need to provide waste materials. There is no decreasing, there's increasing, especially in waste. An increase in the number of red blood cells. Nope, you do not make red blood cells as you exercise. You make new blood cells every 120 days in your bone marrow. Choice 3, an increase in carbon dioxide in her blood. Yes, as you're exercising and you need more oxygen to your cells, you're also producing carbon dioxide, and that needs to leave your body as you're producing it because you're making ATP for your energy, for your muscular energy as you're exercising. So the best choice answer is 3, but let's go ahead and read 4. A decrease in respiration. You're breathing less when you're exercising? No, you're breathing more. Okay. Remember what I said before. When you have a diagram, you want to check out what are you looking at? What is the picture telling you? So this right here is electrophoresis. This is an experiment that takes DNA, okay, and puts it in each of these slots. They're like little pockets. And then electricity runs through this, okay? And then there's a particular enzyme that cuts the DNA in very specific parts, and then it makes these strips. So this is a DNA profile. This is showing you how the DNA will look like after the experiment is done. And then let's say A is the same DNA from the same person as C, then they should have the same exact strips. They don't in this particular case, but I just wanted to give that example. So this is electrophoresis, and this is from Relationships in Biodiversity Lab. So let's take a look and see what they want us to answer. Base your answers to questions 75 through 77. Let me just take a look. 77 is also on the next page. Okay, so I know what numbers pertain to this question. On the information diagram below on your knowledge of biology, DNA samples were taken from 4 different species of animals labeled A, B, C, and D. So this is a different animal than this, or plant or animal. It's an organism. This C is different and D is different, meaning this is your mom, this is your dad, this is you, this is your cousin. These are different organisms. Okay. The diagram below represents the results of a procedure called electrophoresis that separated DNA fragments from each species. Okay, your eyeballs, okay, have to look across, and whichever one, the ones that land on the same pathway, we're going to put a check on them, okay? So let's erase this, and I am going to make, I'm going to make something that would resemble like a loose leaf, all right? And what you're going to do with this paper is you're going to put it across, and this is how you're going to view your strips, your electrophoresis strips, okay? So let's look at the C, okay? C has none in common with any of them. And then let's keep going down. So here we have A, B, and D, okay? Then we keep going down. Then we have B and D. Okay, we keep going down. Okay, we have A, B, C, and D. Okay, let's see here. A, B, sounds funny. D, oh, let me fix that one. It's not working out, guys. Just want to check. Okay. Let's keep going down. All right. Yes, for A and C. Let's keep going down. B and D. Okay. Let's keep going down. None here, none there, none here. All right, guys, let's take a look at number 76. The fragment of DNA separated into these bands because there. So the DNA in this particular experiment in the electrophoresis, once you put your DNA sample in the wells here, the wells meaning it's like an indentation where you can put the DNA in there. Depending on the electrical charge and the DNA coding and the size is the reason as to why they separate the way they do. I'm going to put a video of electrophoresis in the description section because this test is very particular and I want to make sure you understand what this is all about. And it's very difficult to explain this experiment unless you did it and you know exactly what I'm talking about or you studied it. If you've never done this or you really just don't remember, I highly suggest you watch the video that I post in the commentary. Okay? Because I had erased it before. All right. So here we have none. Here we have A, B, D. Doesn't hurt to do it twice so that you remember. These are all good. Okay, moving down and C. Oops. B and D. None here, none here, none here. Okay, so in terms of the most common, okay, here we have 3. Here we have 2. Here we have 4. Here we have 3. Here we have 2. And here we have 2. Okay, so here we have 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 1, 2, 3, 4. Okay. So B has 5 that are similar. A has 4 that are similar. C has 2 that are similar. D has 1, 2, 3, 4, 5 that are similar. So B and D are the most similar to each other because they checked out 5 times together, okay? Then B and 4 B and A are the most common together because they then scored a 4. And then B and C are the least common because they only scored 2 together. And then let me see if there was anything else that I missed. Okay. So now that we know this, let's take a look at how they want us to map this. So the one here, this one that has the closest ancestor, that's going to be B and D because they both scored a 5. So B and D are going to be here. DNA had 4 in common. So D, this could be A. And then the least in common was B and C. Well, let's see, D. D was least in common with C. Okay? Okay, let's move on to the next one. Making connections. Okay. So this one was about the heart, you taking your pulse and then making a graph. This was the bar graph this is what it should have looked like when you finished taking the class results. 78, what is the total number of students who participated in the data collected? Okay, so number of students are right here. These represents the number of students, but you're not writing this. You're taking a look at the bar and where it ended. This one ended at 1, so that's 1 student. This one ended at 2. This one ended at 3. This one ended at 7. This one ended at 4. This one ended at 1. 4 plus 1 plus 7 plus 3 plus 2 plus 1. You add all that together, you get 18. Next one, state one conclusion that can be drawn about heart rates from the data. Okay, one conclusion is there were a lot of students that resulted in a 71 to 80 average. Very little students had average heart rate of 90. Very little students had a heart rate of 50 and below. Those are 3 conclusions that you can conclude there. So I'm going to pick the 71 to 80. The 71 to 80 beats per minute had the highest percentage of students in class. Period. Anything that you can see from the graph, you say, and it would be correct. Okay, number 80. Let's take a look. Let's read together. Base your answer to questions 80 and 81 in the diagram below and on your knowledge of biology. The diagram illustrates the movement of molecules across an artificial membrane in a U-shaped glass tube. A 1% starch solution was poured into the left side of the tube and distilled water was placed to the right side of the tube. Okay, all right, so that's what we're seeing here. Explain why the liquid level on the left side changed over 20-minute period. Okay, explain why the liquid level on the left side changed over 20 minutes. So on the left side, this is left, over 20 minutes, it went higher. That's it. After 20 minutes, the water increased, moved higher up. They're really just explaining what are the observations. They're not asking why. Okay, back to multiple choice 81. Okay. If a 1% salt solution were substituted for the starch solution in this setup, the result would be, I don't think there's enough information to know if salt is going to have any kind of effect, but let's see what choices we have here. Different because all the molecules will move. That is incorrect. Not all molecules are going to move from right to the left because we don't know what it is and we don't know if it fits through that membrane. So it's not one. Similar because a salt would block the movement of It's saying if the salt solution was substituted by starch, so there would no longer be salt, so it's not 2. The same, because the movement of molecules in the tube always goes from right to left. This one makes sense because we're not really getting much information as to why this is moving, but it's definitely not because of the starch. The starch didn't make an effect because the starch just stays here. It's just chilling. Like, it's not because of that. So 81 is choice 3, but let's read 4. Similar, because water molecules will still move across the membrane in a similar manner. No, that's not correct. I don't like this question at all. Okay, this is the first time that I actually see them mentioning a lab, but let's see. During the making connections lab, which is when you took your pulse, several pulse readings are taken and averaged in order to, so let's see. You took your pulse average to get data to see if there are any connections that you can make. Okay, so you took several readings to make sure that you can get an accurate average. All right, so let's see. Did you take several readings because you wanted to get the heart pumping faster? No. You wanted to increase muscle fatigue? No. You wanted to increase reliability? Yes. You wanted to make sure that you were very clear as to what your pulse rate is for the minute. You don't just take one and then, oh, it's 60. No, like, you have to do it several times to make sure you get a solid average. Increase lung size? No. So 82 was choice 3. Okay, number 83. A student using a clothespin beak during the Beaks of Finches lab obtained the results shown below. Okay, so they did the trial. They got these numbers. And then over the course of 4 trials, an average of at least 13 seeds must be eaten in order to go to the next round. Will the student go on? Support your answer. So they're asking, you need a 13 or more in the average to move on to the next trial. So you have to get the average. Okay, so let's calculate it with our calculator together. I wish I could have a calculator on screen. For a lot of you, you'd be surprised as to how many students get this wrong because they don't do the calculations right. So this is what you're going to do first. You're going to add all the numbers first. 12 plus 15 plus 10 plus 11. You're going to put that in your calculator first, okay? As I'm doing now. This gives you 48. That's not your average. That's not your average. You then have to divide it by the number of trials. You had 1, 2, 3, 4. 4. 48 divided by 4 is 12. This does not equal 13 or plus. So, will the student go on? No. The student will not go on because their average was a 12 and did not reach 13, period. Moving on. Okay, guys, ladies and gentlemen, we've reached the end, students, fiber students. And we are at the Beaks of Finch wheel. I can tell you how easy this is. Do not freak out. It's all about practicing. Take a look at the question. Base your answer to the questions 84 and 85 on the diagram below and on your knowledge of biology. Variations in beaks of Galapagos Island finches. Different beaks. They can eat different things. Okay? Some of them are carnivores. Some of them are herbivores. Some of them are omnivores. Omnivores means that they eat both plants and animals. So let's take a look at what they want. If this finch eats plants and this finch eats plants, how is it possible that they're not going to compete for the same? That means that okay, that's fine that they both eat plants, but that just means that they eat different types of plants. So maybe this one eats strawberry plants and this one eats blueberry plants. So they're not competing for the same thing because they're eating different plants. So they may not compete with each other because they might eat different plants, period. Okay. If you're new to my page and this is like the first video you see as opposed to the part 1, just know that like you see how my handwriting is messy. It's not going to be the best because I'm writing on an iPad. My handwriting is actually pretty good in person on paper. You need to make sure that you can read it. Even if your handwriting is not the best, you need to make sure that a teacher can read it, that someone can read it. It's not time to be funny. You're not trying to be funny here, okay? You want to make sure, and at the end, the last couple of, you know what I'm going to do? You know what, just for you? The last question, this one, I am going to try my absolute best at the end of this video, the end of the videos that I'm going to upload for today, for tomorrow's exam. I'm going to do this just for you, just to show you how you have to put effort in the way that you write so that you can be clear all the way to the end. You fight to the end. Let's see the last question. The small tree finch and the small ground finch occupy different niches. This is the role that an animal plays in its environment. For example, bees, their role is to pollinate. Okay, that's a role. The role of a tree is to give shelter, is to feed animals from its flowers and its fruits. Okay, so niche is the role an animal plays. Okay, state one reason other than food why these finches might have different niches. Okay, so how else, other than food, why these finches might have different niches? Why would different finches that eat different foods have a different role? Okay, so just because they're all finches doesn't mean that they all live in the same niche. So here we go with I'm gonna try to do perfect handwriting. Okay, one finch lives on the ground. The other in trees. All right, guys, we are done with this exam. I can't tell you how proud I am of you for finishing all 4 parts, all 5 parts. So if you are here, that means that you did parts 1, 2, 3, 4, and 5. Altogether, it's like 3 to 4 hours or 3 hours, something like that. Congratulations, you practiced an entire exam. I am so proud of you. I am so happy to have been here for you. Online through YouTube. I love that YouTube has this ability. I know you're going to pass. Good luck. Good luck. Good luck. I will see you in the next video. Guys, make sure you subscribe so you can keep getting my videos.